Now I'd like to introduce Eors Szathmary. Dr. Szathmary is a theoretical evolutionary biologist, chairman, head of the Department of Ecology and Plant Taxonomy at Eors University in Budapest since 1995. Also, and equally important, he is a permanent fellow and one of the leaders of the Collegium Budapest, which is an institute for advanced studies in Hungary, in Budapest, that is modeled on the Institute for Advanced Study in Princeton, and they have been doing some very, very creative interdisciplinary work there for the last several years. I had the good fortune to be there for 3 months with Dr. Szathmary, and I have to tell you, on very personal terms, he's a joy to work with and a delightful person, and so it was a great treat for me to be there, and it's a great treat for me to be able to return the favor by inviting him to speak here today. Among his many papers, including a number of great theoretical importance, 2 works where he truly jointly authored, that were truly jointly authored with the eminent biologist with whom I'm sure you're all familiar, John Maynard Smith, are really landmark books in terms of the changes that I referred to earlier that are occurring in evolutionary biology in terms of their understanding of the evolutionary process as a multi-leveled phenomenon. The 2 books that Dr. Szathmary co-authored with John Maynard Smith are The Major Transitions in Evolution in 1995 and The Origins of Life in 1993, which is a somewhat shorter, more popular version of essentially the same volume. Dr. Szathmary also won the New Europe Prize in 1996 at the Institute for Center for Advanced Studies in the Behavioral Sciences at Stanford. It's with great pleasure that I introduce Dr. Szathmary.
Dear Peter, thank you very much for the warm introduction, and I would like to thank for the invitation to this multidisciplinary meeting. As you see, we circumvent the idiot box during this talk, so we are not going to have problems with new technology. Also, I'm especially happy because I am speaking after Professor David Sloan Wilson, and he gave a marvelous introduction to this audience about various basic questions of evolutionary dynamics, and I'm going to use that rather heavily. In the book that was just kindly mentioned by Peter, The Major Transitions in Evolution, we identify a number of them, and here you see the list of all these transitions that we tackle with. Probably it's not all the transitions that we should have dealt with, and Dr. Oro, who is a very leading guy in the field of chemical evolution, suggested that one day I should write perhaps another book with the title The Forgotten Transition, and that should be about the origin of the nervous system. But nevertheless, this is the list of transitions that we are dealing with, and it, as you see for yourself, it goes from the origin of replication until the origin of the human language faculty. And I'm going to explain in some detail what common elements or recurrent elements there are in these transitions. You see a red asterisk associated with some of the transitions, and the asterisk have 2 independent but interconnected interpretations. So one interpretation is the following. We had a small workshop about such problems in Paris a year ago, and I gave this list to the people in the workshop. There were 10 heavy-going experts in evolutionary biology and system science, and I asked them to mark those transitions that they think were difficult. to make. And that is what they did. It was a poll among the experts. And when you look at the list, there is another interpretation. The other interpretation is that, interestingly enough, these are the transitions that we regard today as unique transitions. Unique transitions in the sense that they have happened only once in evolution. Now, here we have to be very careful, because I can only offer an operational definition of that. That the transition is unique means only that all the creatures that have undergone that transition go back to a single common ancestor. It could have been the case that there were other lineages that made the transition, but up to now they are not living anymore. So this is the operational definition we have for a unique transition. But it's interesting to see that people were actually identifying those transitions as difficult that appear to be unique in that operational sense. One could actually ask about the cause of such a uniqueness, and there indeed can be a number of different reasons. One of the reasons that you can give is that the transition is what I call variation limited. That means that the appropriate variation to produce the series of mutations in the broad sense that can actually lead to the transition are very rare, very difficult to make. It's like winning on the lottery, or even more difficult than that, and that gives you a genuine difficulty in the sense that you are not likely to win on the lottery again and again. That's one thing. The other thing is, you could say that the variation that could lead to this transition is not that difficult to make. I mean, it can be rather common, but there is a preemption type of thing, which means that the first lineage to have made the transition is such an overkill to the others who would like to try that later that it just doesn't happen because of competitive reasons. And it could be the case that although the variation would be there, but the selective circumstances that lead to such change are very special. And there is the fourth, that we didn't have enough time. This could apply actually to the broadly defined language capacity. I said that there were some recurrent elements in these transitions that are worthy of mention. And one, I mean, yes, here I have to be specific in saying that it's not the case that all the recurrent elements occur at every transition in the list, but they are sufficiently frequent, so I would like to mention them. So one of the things that you see repeated a number of times is that you have units. Evolutionary units defined as David Sloan Wilson defined them. You have evolutionary units at one level, and then in one lineage at least, it does happen that different units come together and form a higher level whole. And then after some evolution and fine-tuning, then that higher level unit can be treated as an organism in the broad sense, and we shall see example of that. Division of labor, or synergy, as it was emphasized by Peter Corning, actually long before our book, but we didn't know about his book. So I'm happy to acknowledge his work again, because many of the examples that he uses in the synergy book are the same examples that we inadvertently repeated in our book, and that is the idea that a jack of all trades is a master of none, and there are many examples of that. Let me just give you a very simple one. People think that there was a period in evolution when RNA molecules, ribonucleic acids, were not only genetic material but also enzymes. But it turns out to be the case that if you look at it closely, a combination of DNA and protein enzymes is a better solution to this as RNA alone, because DNA is better for information storage and protein enzymes are better for catalysis. This shows you what division of labor or synergy can do to you. This third item is probably a lesser known one, but it is of extreme importance to evolutionary biology. And probably I should dwell more or most of the time on this particular item, because it's not widely known, and popularizers of evolution and molecular biology Even if they know about these things, they seem to miss the point. So when we think about biological inheritance, everybody says it's DNA, 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 and that's it. Now that's not, it couldn't be far from the truth in a funny way. And in fact, there are other type of inheritance systems below and before DNA and above and after DNA that are important in these transitions. Let me give you again a very specific and clear example which everybody will see the force of it. I mean, we multicellular organisms developed from a fertilized egg, from a zygote. That was one cell. And out of that cell, many cells came out, but the cells became different throughout embryogenesis. So they differentiated. For example, they formed a liver cell. The liver cell was not there. The liver cell arose in the process of embryogenesis. But once a liver cell has formed and became to that state, what happens is that when it divides and we do not have liver cancer and so on, it will give rise to another liver cell. So the question arises, where is the difference between the zygote and the liver cell? It's not in the DNA. It is in what we call DNA marking, because genes that are surely there can be turned on and off. That is how we do partly development. But when you actually turn on or off a gene, that state of being turned on or being turned off can be transmitted to the next cell generation. So all multicellular organisms have a dual inheritance system, and the second inheritance system that goes together with DNA has also been called epigenetic inheritance systems. And they are of paramount importance because without such an epigenetic inheritance system, we wouldn't be here. And nobody can deny the importance of such a system, just going from the example. And there is a fourth recurrent item, and again I'm happy that David Sloan Wilson spoke before me, and that is that it seems that for almost all the transitions, local interactions were absolutely important. Local interactions means that you only interact with your neighbors, and that applies to most of your ecology, but also to most of your selection dynamics. And what you see when you want to account for the transitions, that in fact local interactions play a very crucial role. But this is a historical thing if you look at the major transitions, how you can account for events that occasionally were unique. I mean, you don't have a repetition, so what do? You have a sample size of 1, and that is also worrying for life as such. This is why it would be extremely important to find independently originated life on Europa, for example, one of the Galilean moons, but we shall see. We shall see maybe in a few decades, so it's a historical time that we are living here. So how do you make the reconstruction? There are a few methods that you can apply. One is the comparative method. The comparative method includes genetics. You look at the existing organisms and you try to figure out whether the historical transition left any trace of that, or whether a certain scenario can be taken as a hypothesis, and out of that hypothesis certain patterns would follow that you can check whether it is an existing pattern or not. The footsteps of a particular scenario in the biota. The second thing that you can do is that you do computer simulations, and that's very important. And this is through the computer simulations that you discover that the local interactions play an absolutely crucial role. I may or may not get to that because there is a lot of material to be covered, and you can just do it in 1 hour, but 30 minutes is a little bit on the narrow or small side, so we may not get into that. Think of processes that David Sloan Wilson described in the broad sense, and then you are not going to miss this point entirely. It's interesting to see that throughout this transition business, biological complexity did increase. Now, complexity is a very difficult term. It's very difficult to define or measure. There is one definition that I'm using. It's rather clear. It is becoming increasingly better measurable. It's simple-minded, but it is also revealing, and that is that we consider the number of actually expressed genes in organisms. So how many genes are you working with? And that is not genome size. I mean, if there are biologists here, that's not the C value, because the genome contains many pieces of nucleic acids which are not genes, or which we don't know, but for the time being they are not genes, but they are junk or other type of DNA. So we actually concentrate on the number of expressed genes, and we can do that better and better because you have these genome projects and the proteome projects and so on. And if you look at that, then it is true that eukaryotic unicellular organisms like amoeba or paramecium have a higher number of genes than bacteria. Multicellular eukaryotes have a higher number of genes than protists. Animals, as a tendency, have a higher number of genes than plants, and vertebrates have a higher number of genes than invertebrates. And one doesn't know, but probably it would be true that angels would have more genes than vertebrates, and archangels even more genes. You know, it's funny that it actually happened a number of times in recent biology that some oldish ideas, and I can give you many examples, turned out to be for some reason entirely correct. For example, that insects and vertebrates are upside down relative to each other. So what is the belly for me is the back of the insect, and so on. And that was a barmy idea, ridiculed by the Anglo-Saxon world, and it turns out to be true from the point of view of developmental biology. So the scala naturae, as it were, is almost correct if you regard the mean number of genes. It is not an absolute thing, so you can actually find a plant that uses more genes than a multicellular animal, but as a tendency, it's true that you have this increase in complexity defined as the number of expressed genes. And the question is, how do you do that? And one way of doing it is the old idea that was suggested by Susumono, is the duplication and divergence, which means that you start with a gene, then you have a duplication by chance, you have got more copies of that, and since you have got more copies of that, one of them or maybe others are free to drift away in sequence space, as it were. Ultimately, they can acquire a new function, and this is one of the ways how you can increase the number of genes. The other, perhaps more interesting one, is by symbiosis, and that is what I referred to in the introduction, that independently replicating units, ABC, come together. First you have an ecological interaction, and under the right circumstances, which also includes local interactions, it may happen that in fact you find a higher level unit with an increasing rigor of simultaneous replication. I will come back to that. And here is what I already mentioned, that is epigenesis, that you have got a fixed number of genes, but you can work on the genes in this dimension in your epigenetic development. And the asterisk, if you see that, means that here gene A is turned on, here gene B is turned on, here gene C is turned on, and you can build an organism just by turning on and off genes at the appropriate time and the appropriate place. Now, when you consider the transition list that I presented you with, you can ask the following question. If you think of, for example, the origin of the eukaryotic cell, and that's a well-known story because of the eminent work by Lynn Margulis, among others, you know that the eukaryotic cell originated in such a way that some of its organelles came from external sources. They were independently reproducing bacteria like mitochondria or the ancestors of plastids and so on. In that case, it is true that the components that come together are unrelated. They are, ultimately they are related, but they are definitely not coming from the same pool. In other cases, there is a very close relation of the cells. If you want to build a multicellular plant and you start out in such a way that you grow you divide and the two cells stick together, then those cells are almost genetically identical if you forget about somatic mutations. So there the two parties are very closely related. And it was Quelle who, when reviewing our book, noticed that you can actually make a systematic account of the transitions in terms of egalitarian and fraternal transitions, as he calls them. In the egalitarian transitions, it is the transition where initially unrelated components come together, and in the fraternal type of transition, the units that must act together at the higher level are related to one another. And he lists the examples. For the egalitarian type, you have got different replicators in compartments, chromosomes, nucleus and the different organelles, sex. And in the fraternal transition, you have got same type of replicators in cells, same type of organelles, many mitochondria in the cell, cells in the organism, individuals in colonies. Here the units are unlike because they come together from different sources. Here the units are like. Whether you have reproductive division of labor is a good question. Reproductive division of labor is that when there are n number of units together, only a few of them or one of them will reproduce, like in the social insects. Of course, you cannot do that for the egalitarian transition because a plastid cannot do the reproduction for a mitochondrion. That doesn't work. But for the fraternal type of transition, as I have just said, it's a doable thing. Now, you have to control the living together of those guys who are either related or unrelated, but in principle could replicate or reproduce on their own. So, for example, in the egalitarian type of transition, fair reproduction, for example, fair reproduction is enforced by different mechanisms, and mutual dependence is another way to ensure that. In the fraternal type of thing, it is the kinship, as we know from the late Bill Hamilton. I'm very sad to say that he died a few months ago. Kinship is the very important thing that actually can lead to the fact that the unit is not disrupted at the higher level. You always have to start such processes somehow. You have to inquire about the initial gain, because if it is disadvantageous, it will not happen. I mean, let's face it. I mean, natural selection does play a very important role in these processes in the sense that it wipes out all the attempts that are definitely unfit. So there must be some initial gain in the association, and in the egalitarian type, it's the combination of functions, because you see, I mean, in a way, there is already the possibility of division of labor before coming together, because they are different type of organelles or organisms, and they can just fit together so that it is meaningful at the higher level. Here in the fraternal type of transition, it is the economy of scale that can give you the initial gain, which means that 10 lions, although they are closely related to each other, can actually forage or hunt better than 1 or 2 lions, and this kind of synergy at the level of the scale can give you the initial advantage. How the complexity increase is done, here it is done by symbiosis, and here it is done by epigenesis, as also for the social insects. And there is a hurdle, usually, that you have to overcome, and you have to overcome it again and again. Why? Because those units that were once independent still have the potential to behave as outlaws, in the sense that there can be a mutation that gives you the chance to get away from the control of the higher level unit, and we know that sadly enough that happens rather easily. I mean, cancer cells, for example, are those cells that by genetic processes acquire a certain genetic instability, and if this genetic instability is in an intermediate range, you will have a within organism selective advantage of those cells, which ultimately will kill the higher level unit. So the fact the transition has happened is not a safeguard against going back occasionally, and occasionally you go back. There are insects who are secondarily solitary, who have gone from the social state back to the individual state. Why it doesn't happen more often, it's a good question, and it has to do with contingent irreversibility. So control of conflicts, especially when the guys are unrelated that have to stay together, is a very important thing, and a lot of research is being done on that. And if you have heard of such a thing as intragenomic conflict, that very much goes in that category, because occasionally genes behave entirely in their own interest, and that's detrimental to the organism. How many minutes? Another 10, excellent. As I said, one of the common elements is that very frequently in the list at least, a novel type of inheritance systems arose. So let's tackle a little bit this issue, and this will be the final block of the talk. If you want to make evolutionary units, and we want to make them, then they must have the ability to multiply, to have heredity, and to show variation. Heredity just means that like makes like. There is no deeper definition that we need. In fact, Darwin didn't have a deeper definition, and we don't need it either, because if we stick to that kind of generality, we can apply the same theory for many different levels, even non-living ones, which are simpler or more complex than living, once they satisfy the criteria. So the multiplication, heredity, and variability just means that heredity is not always exact. So ultimately, if we are concerned with evolution, these origin of units and so on must go back to chemistry, because it was the world of chemistry that life emerged from. And here is a very simple replicator. It's a molecule which is called glycolaldehyde. Here is a molecule of glycolaldehyde. It's made of 2 carbon atoms and some other atoms. And it's a very interesting reaction, because this reaction has been known since 1861. It was a Russian chemist, Butlerov, who discovered it. So it was not Stanley Miller in 1953 who made the first experiment relevant to the origin of life. It was almost 100 years before that. So what Butlerov did, he took a simple chemical substance called formaldehyde, and he put it in a reaction vessel, normal temperature, mildly alkaline conditions, and he waited. And what he saw was that sugar molecules were accumulating in that solution. There were no enzymes or whatsoever, no living creatures in the test tube. But now we also know the mechanism of it. So what happens is there is a very slow reaction that out of the 2 formaldehyde molecules gives you 1 molecule of glycolaldehyde. And the glycolaldehyde takes up another formaldehyde molecule. You have a compound of 3 carbon atoms, another formaldehyde. And what happens here is very interesting, because this tetrose, a sugar made of 4 carbon atoms, spontaneously falls into 2 molecules of glycolaldehyde. Such processes are called autocatalytic reactions, and here is the abstract form of the autocatalytic chemical cycle. Now, such molecules can multiply. The question is whether they have heredity. It's a very open question whether such simple chemical systems can have heredity, which means that you could have A-type cycles, B-type cycles, C-type cycles, and when they would do the autocatalytic increase, then the A-type would give rise to more of the same, B-type to more of the B, C-type more of the C. But I am actually I strongly think that such a phase of chemical evolution was there, and in fact simple chemical autocatalytic systems were the first replicators. And variability means that occasionally, by sheer chance, one intermediate of cycle A transforms into intermediate of cycle B, and there a new variant can arise. Just to give you the full complexity of the Formose reaction, this is all the interconversions that we know of, but the hardcore is still this autocatalytic part. That is what, when you didn't know what complexity was, then it doesn't matter, you see. No, chemical space is not in, the networks of chemicals are not in the same, in ordinary physical space. But anyway, you can appreciate the complexity. The question is how you get from such a thing that may or may not show heredity, but even if it shows heredity, it can show only so-called limited heredity. And that is a very important concept, and I would like to say a few words about that. The DNA that we have today in our genetic material can give you an indefinitely large number of different genomes. Why? Because of sheer combinatoric reasons. If you take, for example, all the possible DNA molecules up to a length of, let's say, 100. This is a very small molecule. It's just about the size of transfer RNA molecule. So imagine that you want to make, or let's take a virus that's an existing thing and it replicates when the cells are around. Viruses can have 4,000, 5,000 nucleotides, okay? So let's try to see, let's try to build a museum in which there is one copy of each of the possible sequences of length 4,300, let us say. And you will figure out very easily that there is not enough matter in the universe around that you could build a museum which would show only one specimen of all the possible sequences. So present-day DNA-based system has such an importance because it is potentially indefinitely large, the space that you can explore. Whether you explore it or not, that's a historical thing. Now that cannot be true, of course, of systems like that because they have limited heredity, which means that the number of types is in fact much smaller than the number of molecules that you can actually have in a pond. For DNA, it's the other way around. The number of types is much larger than the number of individuals that you can have in a realistic system. If you want to make the transition from the non-living to the living, or from lower level units to higher level units, you should be able to make the important transition from limited heredity to indefinitely large hereditary systems. And that is true for two other systems as well, and by this I would like to close the talk. One system where it is again apparent is epigenetic inheritance. Now, I said that without the ability to make heritable changes to cell types, we wouldn't be here because the multicellular organisms couldn't have been formed and couldn't be maintained. That's fine. The question is, where does such a system come from? Of course, we originated from protists, creatures like paramecium and amoeba. The good question is whether you can find systems like that there, and you do find systems like that, and in fact, since they are unicellular organisms, the hereditary changes that are epigenetic in them carry over to the next generation because the generation consists only of one cell. So there is a molecular Lamarckism involved in those systems, but only a few number of states can be maintained by the rudimentary epigenetic systems of amoeba. A few cell types can be propagated, and that's it. That's not enough for us. So during the origin of multicellular life, limited hereditary epigenetic system must have given rise to much more powerful epigenetic hereditary systems, which allows to the specific formation, the maintenance of at least a few hundred cell types, depending on how you count cell types. Davidson would probably say that the cell types that we actually do work with are in the realm of 10,000, because you have to count them a little bit more carefully. And the last item I would like to mention is about the last transition, and that is about the origin of the human language faculty. The human language faculty is undoubtedly how should I say, genetically preconditioned, in the sense that without the appropriate genetics you cannot speak. But of course the converse is also true. With the appropriate genetics, it's not a guarantee that you will speak. The appropriate environment is absolutely important for that. So it's actually one of the neatest interplay between complexity and genetics that David Sloan Wilson was referring to. But the language as we use is the basis of cultural inheritance, and it is an indefinitely powerful system in the sense that an indefinitely large number of statements can be made by the same generative mechanism. That's fine, but we don't see that in animals. What we see in animals at most is that they can make a finite number of different statements in double quotes, and you must have, and that is what we call proto-language, and you must have made the transition from a very limited system, which is called proto-language, to a system where an indefinitely large number of sentences can be made and can form the basis of unbounded cultural evolution, of which one of the results is that you can actually talk about such things in a Congress of System Science. So, thank you very much.